would like to thank the conference organizers for inviting me to present this research. So, seeing as how PEth is an objective measure of alcohol consumption, several labs have developed bio-analytical assays to quantify this analyte. Right now, PEth is primarily quantified via LC-MS, and many groups extract this from whole blood. Regarding dried blood spot or DBS-based methods, there are a limited number of labs that have published DBS-based assays, and in the United States, many research groups utilize the United States Drug Testing Laboratory, or USDTL, to conduct their PEth testing in DBS. So our lab, the Colorado Antiviral Pharmacology Laboratory, was also interested in developing a DBS-based PEth assay for a couple reasons. To begin, we primarily do research in individuals with HIV and hepatitis C, and we were interested in quantifying the effects of alcohol consumption on disease and treatment outcomes in these populations. For our studies, we typically collect dried blood spots every few weeks, and we also have a lot of experience developing DBS-based assays, so this PEth one was ideal for us. But also as a matrix, dried blood spots have a lot of advantages over whole blood. To begin, they have small volume requirements, which makes them easy to collect, transport, and store, and they've also oftentimes been seen to improve sample stability, and this has been shown in the literature with PEth as well. So we went ahead and developed a DBS-based PEth assay, and on the left here you see our standard curve, and the way that we make our standards is we obtain blood from individuals who do not consume alcohol, and we spike it with known concentrations of PEth. Our curve goes from 5 to 500 nanograms per mil, and if you look on the right here, you see a table of proposed PEth concentrations associated with different types of consumption based on Olwelling's comprehensive review of PEth literature. So our standard curve is definitely encompassing this range. However, we have seen PEth concentrations in the thousands in literature, so we did validate dilutions of up to 16 times with our assay and now have the potential to quantify PEth up to 8,000 nanograms per mil. And for some more validation data, we found that our assay was accurate and precise across a variety of spot volumes and dilutions. We also did a variety of stability testing, and the data that we found was really promising, and we will continue to add to this through long-term assay maintenance. And then we also found that things like carryover and crosstalk, the presence of commonly used concomitant medications, and matrix effect had no significant impacts on PEth quantification. However, we do realize that dried blood spots are a unique matrix, and with that comes a lot of factors that may impact analyte quantification. So we looked at those in our validation as well. And one of the big things is the hematocrit effect. And this is based on the observation that individuals with higher hematocrits will have smaller, denser dried blood spots. And this becomes an issue when you are taking a fixed area or punch, because you would be extracting higher volumes of blood and inflating someone's PEth concentration. So we found that our assay was not immune to the hematocrit effect. Here you can see the differences in PEth concentrations when going from a 30 to 40 to 50% hematocrit. However, because we were able to identify this, we did adjust our method accordingly, and we now create our standards from blood with a hematocrit of 42 to 45% for two reasons. The first is that this is about the population average, so we're essentially eliminating the hematocrit effect from a vast majority of samples that we may see. But also, this is in the middle of that physiological range, so we're lessening the variability on either side. Now, during our validation, we were approached by two investigators that are part of the NWCCS. And this is a research effort that aims to understand chronic health conditions affecting people living with HIV. To date, this is the longest running observational HIV study and has over 4,000 participants. So these investigators approached us with the opportunity for collaboration in which we would be quantifying PEth in their samples. And we were really excited about this, and in preparation for it, we asked if they could send us samples that they had previously quantified through the USDTL so that we could do a cross-validation. And we had two objectives with this. The first was to assess our PEth assay using patient samples with authentic PEth, and the second was that in doing this cross-validation, we would be able to assess the interlab reproducibility of PEth quantification. So they agreed, and they sent us a combined 46 samples from two different studies. 25 came from a study called ADEPT, which took place from 2012 to 2015 and was collected in Uganda from persons with HIV. And 21 came from a study called HCV Cure, which took place from 2018 to 2020 and was collected in the US from persons with hepatitis C. In addition, this table here is showing some differences between the assays that our lab has and the USDTL has. So for the reference material, they utilize Enzo Life Sciences, whereas we utilize Cerilliant. For the standard curve, theirs goes from 8 to 800 nanograms per mil, and ours goes from 5 to 500. But again, we can quantify up to 8,000 with those dilutions. And then it is unknown to us through the publication of the USDTL's method whether they did any DBS-specific testing, whereas we did assess for some of those factors. But ultimately, we hypothesized that cross-validation data would show that PEth quantification by our labs would be in agreement, and thus we would establish interlab reproducibility. So for our methods, in terms of PEth quantification, both our lab and the USDTL utilize LC-MS-MS, and we both quantified PEth 16.0.18.1. After our lab quantified PEth in those cross-validation samples, we did something called incurred sample reanalysis, or ISR, and this is a quality control measure to ensure the reproducibility of our data. What we did was we re-extracted and quantified PEth in 30% of our samples, and then found the percent difference between repeated and original concentrations using this formula here. An acceptance criteria for ISR is that the percent difference be less than 30 for 2/3 of the retested samples, though we also assessed our data with a 20% acceptance criteria as well. And then for the comparison of cross-validation data, we looked at the Spearman correlation, we looked at the R-squared and slope of the trend line that comes when we plot our data versus the USDTLs, and we also looked at the percent differences in concentrations. So moving on to results and beginning with our lab's quantification of those cross-validation samples, you can see that we had a wide range of concentrations all the way from BLQ, because it was below 5 nanograms per mil, to 1600 nanograms per mil. And of those 46 cross-validation samples, 17 were BLQ and 7 were originally above the range of our curve and had to be re-extracted with dilutions. And looking at our incurred sample reanalysis data, you can see that for these 14 samples, when we compare the repeated to the original concentrations and then look at those percent differences, 100% of our samples meet that 30% acceptance criteria. If we want to assess this using the 20% percentage instead, we have 2 samples that just fail that, but that still gives us an 86% pass rate, which is above the 67% needed. So our ISR was successful, and we were confident in the data that we had produced. Moving on to the cross-validation results. So what I've done here is I've plotted our cross-validation data by study that was sent to us. And on the y-axis, we have our lab's concentrations, and on the x-axis, we have the USDTL's concentrations. And right away, looking at these data, the first thing you notice is that we are seeing very high correlations, with rows of 0.97 and 0.99. However, when we look at these slopes of these trendline equations, we start to see the difference in quantification, which is that our lab is quantifying PEth 1.5 to 1.7 times higher than the USDTL is. And when we see this difference, the question becomes, is this happening in a certain concentration range, or is this a more consistent effect? So to answer that question, we looked at the percent differences in PEth concentrations per sample. And here I'm just showing the HCV cure data, but know that the depth looked almost identical. So when we compare the USDTL's concentrations with ours, and we look at those percent differences, you see a lot of zeros in this last column. These are from samples that we both identified as BLQ. In addition, there is a 100% different sample, and this is from a sample that we quantified at 6 nanograms per mil that was BLQ for the USDTL. However, when we look at samples where we were both able to quantify PEth, you can see that whether we are looking at low, mid, or high concentrations of PEth, we are seeing this consistent 30 to 50% difference in quantification. And ultimately, across all cross-validation samples, we were seeing this median difference of 36%, with our lab having those higher concentrations. So the question becomes, why are we seeing these differences? And we looked through literature to try and identify some of these sources. And one of the things that came up was Regio isomeric purity. So the CAMAC DBS lab conducted a study where they wanted to see if the Regio isomeric purity of PEth reference material impacted the quantification of unknown samples. And what they found when comparing Cerulean, Avanti, and Chiron reference material was that Cerulean had the highest Regio isomeric purity, mimicked what was seen in biological samples, and did not significantly impact PEth quantification. However, Avanti and Chiron reference materials had impurities within them that were causing a significant over or underestimation of PEth concentrations. So our lab uses Cerulean, and this paper was actually a big inspiration for us to make that switch, but the USDTL utilized Enzo Life Sciences. And though this isn't a vendor that was tested in this study, we now know that unless it has a Regio isomeric purity that mimics what was seen in Cerulean, this is going to be a potential cause for those differences. In addition, it is unknown if there are any DBS-specific factors that may have caused these differences that we're seeing. So, for example, based on the Hematocrit Effect data that we found in our validation, we hypothesized that if the standards by our lab and the USDTL were created from blood of different hematocrits, then this could also be a source for those differences. Really nicely though, there was also a study where the CAMAG DBS lab's PEth concentrations generated via their automated method were compared to concentrations generated via the USDTL's manual method, and their data looked very similar to ours. So this is a figure on the left from their manuscript, and on the y-axis is PEth concentrations via their automated method. On the x-axis is those from the USDTL. And the samples used in this study were from ADEPT, so here I just once again included our ADEPT cross-validation data so that we can compare. So again, we are seeing very high correlations with the USDTL with rows of 0.98 and 0.99, but in terms of the median PEth concentrations, we were both quantifying PEth 1.6 times higher than the USDTL was. So in conclusion, our lab developed a DBS-based assay and we validated it for both traditional and red blood specific factors. In addition, our assay was shown to be accurate and precise when quantifying PEth in patient samples as a result of that successful ISR. And doing the cross-validation with those samples previously quantified by the USDTL, although we were seeing high correlations between our data, our concentrations were higher than of the USDTL. But these findings are consistent with the CAMAG DBS lab. And then for future directions, we believe that the potential sources for these discrepant results does require further study. Because again, though we were seeing high correlations between our data, differences in exact PEth concentrations could lead to interpretation differences regarding the extent of someone's alcohol consumption. And we believe that if we want to maximize PEth's potential as a quantitative biomarker, we should be aiming for that interlab reproducibility. And with that, I would like to thank Dr. Jennifer Kaiser, my PI, for all her support and the opportunity to work on this project, as well as the entire CAVP lab. Drs. Judith Han and Phyllis Tian for supplying us with those cross-validation samples, and these are our funding sources. With that, I'd be happy to take any questions.
Thank you, Lana. So, are there any questions? As you can go.
Thank you. Indeed, nicely showing the importance of interlaboratory comparisons. In your methods, did you evaluate whether the dry blood cell concentrations are the same as if you would measure them in liquid blood? Because we find that that's something which is very important to do as well, that the dry blood gives the same result as the liquid blood.
Yeah, I agree that that's a very important study. We do not have a method to quantify PEth in whole blood, so that's not a comparison that we've done, but that would definitely be something interesting to look at, if those quantifications would be the same.
Okay, thank you.
Are there other questions or comments? One more.
I have a question about, sorry, I'm looking at key research. That ISR study, so using the same spot or same count? How do you consider the sample in the ISR?
So it's not the same spot because when we extract with the spot, we then discard it later. So it's samples, it's from the same card. So it's spots that were taken at the same time, but we're just using a separate one.
Thank you.
Yes.
One last question.
It's just a comment. The other thing, the other difference between the USDTL and the test you did was probably about 5 years,
Yes.
which is kind of amazing.
Yes.
They were in storage for that long.
Yes.